Hello everyone, hope all are fine. Today our class 10 biology class is not about something only based upon your NCERT. Or not that here from you are getting means on the next day 11th examination. Rather here we will talk about practical life experiments. Whatever the topic we are having, based upon that, but in an extended way. Let's have a look. Chapter 6, Life Process. Here from we will gather some practical activities and some experiment details. Let's have a look. What we are saying here? A leaf that is attached with the plants. A black paper strip is attached here, so that these parts are not getting the direct sunlight. So here will be, yes, no starch. These parts are having the starch. That's why this is the area of leaf. If we add iodine to this, because iodine is the iodine test, is the best test for the starch, right? So if we add iodine here, this part will not take the color, rather these parts are taking the color. So this is the areas of leaf affected with iodine, the lower and the upper half, middle half without iodine as no sunlight. Next part. So here we are having few leaves. Here the leaves we are seeing that starch free leaves. If we attach the black paper, then in the alcohol and in burner we are going to do the boiling. Chlorophyll removed mostly. Then if you add iodine, this triiodide form, which imparts a blue-black coloration with the starch. That you can see, upper half and the lower half is blue-black, like the first figure. Middle half is the green. What's next? This kind of coloration you will get in reality. The blue-black coloration in the second leaf middle, and this is first one before the experiment, before the addition of the iodine. What's this? The next set of experiment, right? So let's talk about it. Here a plant leaf is covered within a bottle which is having potassium hydroxide, KOH. Potassium hydroxide is having the capability to absorb the CO2. So, it will absorb all the CO2. Now, it's like this, KOH plus CO2, potassium bicarbonate. So, again this lower half is not getting here the CO2, it can't prepare starch. Next diagram. Here you can see the lower half with the no starch. In the iodine test, no coloration, rather the greenish color will persist, blue-black coloration in the starch. Here, as which part was outside of the bottle. That will do the photosynthesis as it has done the photosynthesis. So now it takes the color of the iodine test, and this part, lower half, it is not taking. So let's move on. Next experiment. Okay. Next one, this. Here you can see if oxygen is not available, it will continue to respire anaerobically, but in doing so, it receives much less energy. That experiment I have already shown in the last slide. So, glucose, a little energy, carbon dioxide and alcohol. This is the experiment. Equation: CO, calcium hydroxide, which will react with water to prepare calcium hydroxide, that is lime water. Now, CO2 with that will turn it to calcium carbonate, that is actually milky white. I am again repeating, calcium hydroxide with CO2 it will turn calcium carbonate. That's milky white. Now, when more CO2 added, it will form calcium bicarbonate. That is actually colorless. So here we have seen that this particular structure diagram that turns almost milky. Test for CO2. Carbon dioxide turns lime water cloudy. CO2 plus calcium hydroxide, that is calcium carbonate and water. Okay, so let's move on. Now, this is a very funny experiment. Sugar yeast experiment which you can easily do at home. You have to buy some yeast powder. You take a bottle. There you can add some sugar solution, fruit juice or honey solution or honey. There you add the yeast powder. Now the CO2 will evolve and that will bulge out this. It's like a magical experiment. The sugar yeast experiment. As more anaerobic respiration there, then more bulged out of this particular balloon. Or if less amount of glucose is there, less amount of this bulging out. So that's the sugar yeast experiment. Or you can do it with the cake preparing powder, that baking powder. With that also you can do it. So that's it. It's very interesting. So let's move on. Now this is nothing but the testing of the salivary amylase. How it is going on? Salivary amylase is a kind of the protein. In the right side we are seeing the protein structure which is having the alpha, beta, this kind of polypeptide chain. Without enzyme, you will take one, that's a control experiment, and with enzyme, that's the experimental setup. Actually that is the procedure in what way we perform experiment in the lab. And this one for the temperature provider, hot plates. So here we are taking starch solution, one without enzyme, one with enzyme. Now if I ask you, if you are going to do this experiment, how you will do? You will buy enzyme? No. We are simply having the salivary amylase in our mouth. When we eat something we like, then our mouth waters. This is actually not only water, but a fluid called the saliva, secreted by the salivary glands. Right? Those salivary glands are secreting these polypeptides, proteinaceous, those salivary amylase. Now we will make one setup without this waterly secretion, without, and one with this. Now if we add starch is there, and after that if you add the iodine, of course one will take the color of the iodine bluish back, right? Because starch is there and one will not, as starch is broken. If starch broken, if starch already broken into the smaller component, then no sugar monosaccharide, those things are there. So iodine can't bind there, that will not take the color. And where without enzyme, starch intact, so it will look like this. Test for starch in glucose. Add starch 2 ml and saliva to clean test tubes. Swirl to mix and let them sit for 5 minutes. Easily you can do. Iodine test on the starch here 1 and 2 for the Benedict. Here you come to know another solution, that's the Benedict reagent, where we can heat for 5 minutes. Again I am repeating, it's not needed for your exam, it's for to know, to gather information. Okay, how the labs are working. When we are talking about how your enzymes are working, how the lab test, how they are doing or how you are supposed to do in the higher classes. So Benedict reagent is there, heat for 5 minutes if there. So then how it's changing? If iodine solution is there, starch saliva mixture, there will not such kind of coloration, and if Benedict reagent, it will form a particular PPT which is visible from the outside. Let's have a look in the next slide how it works. Okay.
In this investigation, we'll use an enzyme to break apart complex starches into simple sugars.
We'll use an iodine solution to test for the presence of starches. Normally, this solution is this yellowish color, but when this solution mixes with starch, it chemically reacts, becoming a very dark purple or black color. This makes it evident that starch is present in the solution. Our iodine solution has been used to show that both of these test tubes contain starch. To one test tube, we'll add an enzyme that will digest these starch molecules.
The enzyme we'll be using is called amylase. It's the same enzyme that's found in your saliva, and allows your body to start digesting starches as soon as you begin to chew your food.
We'll place both of our test tubes into a warm water bath. This will allow our enzyme to work much faster.
Iodine makes the starch solutions black, but once the starch has been digested by the enzymes, that black color will go away.
You can see here that after our enzyme was allowed to work, that's exactly what happened, and the color in that test tube went away.
Now that the enzyme has digested our starch, we'll test to see if it was broken apart into simple sugars. To do that, we'll use Benedict solution. Benedict solution tests for simple sugars. When sugars are placed into this solution and heated, a precipitate is formed. That precipitate provides evidence that simple sugars were present.
We'll take some of our digested starch and place this into our Benedict solution. This will allow us to test for the presence of sugars. Then we'll place our solution over a flame so that it can be heated and observe whether or not a precipitate is formed. It's very important that whenever you heat the contents of a test tube, you take the proper safety precautions. Here you can see the precipitate at the bottom of the test tube.
So there you have it. This lab investigation provides evidence that when starch, a complex carbohydrate, gets broken apart into smaller molecules, those smaller molecules are in fact simple sugars.
So, Phil, you understood this? So many this kind of experiments are available. I shared here only one. So, now I will move on to the next part. Let's have a look. What's this? Perhaps we have gone through this one. Manometer, the instrument. Many times your parents or you are going to the doctors for this purpose, that whether your pressure or the elder's pressure are normal or not. So, first, this is the artery, brachial artery, normal blood flow, no occlusion of the blood flow. If the cuff is there, this is a cuff, right? Pressure blocks the blood flow. Now, you can measure the systolic pressure. Which one is systolic pressure? When your ventricles, they are in the contracted form. Then when you start release the blood, that's the systolic pressure. That's the cuff pressure. Your doctor, they can hear it. And diastolic pressure, cuff pressure, and that means can't hear. It's very much less. That means systolic pressure, that's we are talking about the higher blood pressure. So, the pressure of the blood inside the artery, during ventricular systole. Contraction is called systolic pressure and pressure in artery during ventricular diastole relaxation is called diastolic pressure. The normal systolic pressure is about 120 of He and diastolic is 80 mm of He. Here we are seeing that how with the sphygmomanometer we can measure the pressure. Let's have a look if you are given or if you are going to measure how you will. So let's take a look.
Hi, my name's Simon. I'm one of the final year medical students. Can I just check your name and age, please?
Yeah, my name's Andrew and I'm 25.
Nice to meet you, Andrew. Today I'd like to take your blood pressure, so that will entail me taking this cuff, inflating it around your arm, feeling and listening to your pulse. Does that sound okay?
Yeah, that's fine.
It might be a little bit uncomfortable, but it shouldn't hurt.
That's alright.
Are you okay for me to go ahead?
Yeah.
Lovely. If you just pop your arm on the table, that's lovely. Fine. And I'm just going to feel your pulse now. That completes the procedure. Thank you very much, Andrew.
Thank you.
To complete the procedure, I would check the blood pressure in the patient's other arm. I would then record the results in the patient's notes.
So hopefully you understood regarding this kind of extra knowledges or extra things, and I think you liked it. In next class I will come with this kind of videos more. If you like, have a talk with me so that I can give you so many this kind of things. Okay, thank you. Have a nice day ahead and we will meet and enjoy in the next class. Thank you.